Good morning, everyone. Welcome to today's webinar on Sclerotinia in canola. I'm here with Rajat Kangura, our canola specialist with the Department of Agriculture and Food in Western Australia. So good morning, Rajat.
Good morning, Dominique, and good morning, everyone. This morning, I'll talk about implications of Sclerotinia in the current growing season and how to manage it. I'm sure all of you are enjoying one of the best growing seasons in the century, but at the same time, I think that fear of Sclerotinia might be lurking on our minds. So this morning, I'll give you a quick snapshot of the current situation of Sclerotinia as far as where we are at the moment, especially in terms of the current conditions. And also, I'll give you some of the information about the problem that we are having, especially Sclerotinia infecting pre-flowering. The disease outbreaks are usually favored by continuous wet conditions and humid conditions during flowering time. We have seen numerous reports through pest facts and through personal communication via Twitter, emails, that apothecia have formed or emerged in a lot of canola growing areas of Western Australia, and usually this is actually, as I mentioned, favored by cool, humid, and wet conditions in most part of our wheat belt. So, what options we have in terms of managing Sclerotinia? Research over the past 5 to 6 years have shown that fungicide are the main tool for us to control Sclerotinia in season, especially in the events of early epidemics. The best timing for fungicide application is at 15 to 30% bloom. And in the event of late epidemics, which happened in 2013, in particular in our northern ag region, in that situation, the fungicide application was quite effective when targeted at 50% bloom. So what we have seen in our southern region, where usually the disease epidemics happen early in the season, the fungicide targeted at 25 or 50 percent stage gave us the best control of the disease and also give us the best economic return on our fungicide application. Okay, so why should we worry about Sclerotinia too much? Why this is so important? I should mention that Sclerotinia is the most destructive disease everywhere wherever canola is grown, and especially in Western Australia. So it can cause a lot of yield losses, especially in 2013. We estimated that Sclerotinia cost us about $50 million in lost production. And in 2014, these losses were almost half due to unfavorable conditions, especially during flowering. And in some worst affected crops, which are heavily infested, the losses can be up to 40%. So this clearly shows us that we should be on top of this problem in order to reduce production losses from this disease. Okay, so this is just a quick snapshot what we have found so far in the current growing season. As you can see, the apothecia have almost emerged in most of the canola growing areas in the northern, central, southern, and south coastal regions. As you can see, all those names of the towns where we found these apothecia. And in some areas, especially few crops in the northern region and many crops in the central and southern region have actually shown signs of pre-flowering infection. Okay, so I'll just quickly go through the disease cycle of Sclerotinia. I'm sure most of you know what exactly the cycle starts and how does it happen. So, for example, the traditional disease cycle of Sclerotinia starts with the emergence of apothecia from sclerotes, then it leads to petal infection, leaf infection, and then finally the stem infection. So this is the main predominant disease cycle. But there is another type of cycle which is usually not very common. It starts with the direct germination of sclerotes without the formation of apothecia. And these sclerots, when they germinate, they form strands of hyphae which infect the lower leaves, and from lower leaves through the petiole, the infection gets into the stem, and in case of severe infection, as you can see here, it can kill the entire plant. So what the general belief was, in the past we were thinking that this type of disease cycle is of rare occurrence. It can happen, but it wasn't very common. But for the last couple of years, especially last year and more so this year, the this type of cycle is on the rise, and we are seeing lots of crops which have been infected by direct germination of sclerots and causing a lot of stem infection as well. So I would really encourage everyone to go out and scout your crops before they start flowering, and notice if they find any pre-flowering infection. So these are some of the management options that are available for managing Sclerotinia. For example, that includes cultural practices that involves long rotations with non-host crops, using seed free of sclerots, using wide row spacing to reduce the airflow through the canopy, and also deep burying, if possible, to bury the sclerots quite deep into the soil so that they aren't able to germinate and produce apothecia. But all these cultural practices has to be applied before the start of the season. So now we are pretty much in the middle of the season now, so we can't go into the reverse gear. So what options is available for us is to use fungicides, of course. So some of the products which are registered for the control of Sclerotinia include a number of products which you have active ingredient including prothioconazole and tibuconazole, some products with prosimidone and iprodone active ingredient. What is most important for using fungicide is, of course, the timing of application, which is absolutely crucial in order to get maximum benefit of fungicide sprays for controlling Sclerotinia. Okay, so now, as I said, Sclerotinia is now on our doors, and we surely need to decide very quickly how to manage this problem. So you need to consider the key risk factors which determine the occurrence of Sclerotinia in your crop. And these risk factors include, first of all, is presence of inoculum in your current canola crop or any paddocks surrounding your current canola which had a history of Sclerotinia in the past. And secondly, you need to be aware of your bloom stage of the crop, and especially if the crops are at very early stage of flowering and they are at the highest risk given the current conditions of developing Sclerotinia later in the season. And thirdly, the plant density. If your crop is very dense, and surely the risk of developing stem infection is very high. And then also it also depends upon the seasonal conditions, especially the conditions before the emergence of first flowers on your crop. That will actually determine if the apothecia have been produced or not. And secondly, favorable conditions are also required post-petal infection, especially for the first 3 weeks. The conditions have to be very wet and humid for the disease cycle to continue in the crop. Once you have considered all your risk factors, and then you have to weigh up your yield potential of the crop, cost of fungicide application, and the fungicide itself, and also the current canola price. So all these other factors which you know probably by now, one of the hardest thing is you have to find out what are the conditions which will drive the epidemics in your canola crops. So to address this, we have done a lot of work, especially over the last 5 years, to find out the major weather drivers which are responsible for causing a lot of disease in your crop. So with the result of petal testing over the last 5 years, so we have found the triggers. I'm very pleased to say that. So those conditions involve if you have more than 40 millimeters of rain over the 3 weeks preceding the emergence of flowers in your crop, and with the more than 75 relative humidity. So that means the apothecia are produced and they are releasing spores. And immediately after that, if the outlook is for wet conditions, similar conditions, more than 40 millimeters of rain coupled with more than 75% of relative humidity over the next 3 weeks, that will drive the disease epidemics in your crops. Okay, let's talk about the most vulnerable stage of the crop, when to apply fungicide application. So how to identify different bloom stages. So basically what you have to do is count the number of flowers on the main stem. So once you count, you have to count the open flowers plus the pods as well, and also the pods that have aborted. So if you look at on the left hand side of the column, so this is percentage bloom, and the corresponding number of flowers that should be open on the main stem is on the right hand side. For example, if you look at the 30% bloom, the main stem should have at least 15 to 20 flowers open on the main stem, and be aware that this all includes the open flowers, also the pods that are formed, and also the pods that have aborted. So this will give you an indication what growth stage or what bloom stage your crop is. So what happens after 50% bloom? The petals that have already formed, they start falling, and at 60% bloom you will see the yellow color starts to fade, and at about 80% bloom you will see there were only there most of the pods on the main stem and branches, but the number of flowers has significantly reduced on the main stem. Okay, so uh, now is the time for you to consider all those risk factors which I have already mentioned, and apply fungicide at the appropriate bloom stage. To help with that, I would like to give you an example from one of the fungicide spray timing trials that we did in South Sterling's last year. So this trial was actually done in a very high disease pressure where canola was like 1 year out from rotation. The trial was done on top of 2013 stubble. So various spray timings that we used for this trial included fungicide targeted at 25% bloom, 50% bloom, 60% bloom, and then we also included two spray applications, one early and one mid bloom stage, which was 25 and 60% bloom, and also very late infection, very late application 3 weeks after 60% bloom, because we are often asked a lot of questions, once you have passed 50% bloom, whether it is worthwhile for you to apply fungicide or not. So to address this question, we included this treatment in the trial. So I was very glad with the outcome of the trial. As you can see here, all these fungicide spray timings were very effective in reducing the disease levels compared with the nil. As you can see here, very high level of disease. But the treatments where we applied fungicide at 25% bloom or 50% bloom were surely most effective compared with the other treatments in reducing disease levels and also improving yield. As you can see here, this is the yield of nil plot, and this is the yield which was about 1.6 ton per hectare with 25% bloom application, and it rose to about 1.75 ton per hectare, and so on. As you can see that the only thing which I think I must mention here, that when we applied fungicide very late, 3 weeks after 60% bloom, surely there was absolutely no effect of that late application. In terms of two application, surely they give as good response as just one application. So it really reinforces my previous messages that well-timed single application will give you the best control of the disease, and you will get the best economic return by this application. Okay, so let's look what sort of economic returns you got with all those spray timings. As you can see, the best return on investment was when we applied fungicide spray at 50% bloom. So this is because we applied the spray when maximum number of petals were open on the main stem. Although we got very good response with 25% bloom application, but surely when we apply to cover a lot of petals, so that's where we got the maximum response. 2 application, not as good as single application. You didn't get much out of 2 applications last year. So which shows that last year the second application wasn't really needed given the prevailing weather conditions at the time. So we got a little bit return with a late application, 60% bloom, but really the main highlight is 3 weeks after 60% bloom didn't give us any benefit. In fact, it chewed up all our cost of fungicide application. Okay, so now you know at what stage we are in the season in terms of Sclerotinia disease progression, and you can use this example which I have just shown you as a guideline to make a decision whether you want to apply fungicide and when you want to use fungicide application. And you can utilize the rule of thumb which I said, uh, the conditions, the main weather drivers that will drive the epidemics in your crop. If you are not sure the weather condition over the past few weeks, you can look up the weather stations, especially the map from DEFA, that will tell you what sort of rainfall you got over the last few weeks, and look at the BOM forecast, what is going to happen in the next few weeks. So surely all these factors, I'm sure, will really help you in making your decision when you want to apply the fungicide application. And I'm sure this year it will not be difficult to manage Sclerotinia this season. So for further information, you can refer to all those links that I have listed here if you need to get more information on Sclerotinia management. Thank you for listening, and thank you to Dominique for organizing the webinar, and also thank you to GRDC for supporting this research.